Dear learners, I welcome you to this module which will help you to understand the structure of DNA. The first question which arises is what DNA is. DNA is a type of nucleic acid, or we can say that it is a type of molecule which is big in size, and therefore it is a type of macromolecule. So there are various types of macromolecules like carbohydrates, proteins, or lipids. In the same way, nucleic acid is also a type of macromolecule. Now, this macromolecule or the nucleic acid which we are talking about is divided into two types. The molecule are of two types, right? One type is what we are studying, that is DNA, and the full form is deoxyribose nucleic acid. And the second type is called as RNA, which is ribose nucleic acid. Okay, so here the term comes deoxy, that is one oxygen is less, and here it is ribose, means no deficiency of oxygen. That means, let us write over here that oxygen is present. Okay, so now we have a concept that the nucleic acid are of two types, deoxyribonucleic acid and ribonucleic acid. One more very distinct feature is that the DNA is a double-stranded molecule and RNA is a single-stranded molecule, right? There are only exception are viruses, that there are many viruses in which DNA is present in single-stranded form and RNAs are present in double-stranded form. So the viruses, some of the viruses are the exceptions for this rule. Otherwise, DNAs are double-stranded and RNAs are single-stranded molecules. Okay. So the first structure which was widely accepted and recognized, which explained the structure of nucleic acid, was proposed by Watson and Crick in year 1952. The which was proposed by Watson and Crick was called as double helical model, that we will come at the end with some of the statistics of the model. The model first proposed that DNA is actually a polymer, okay, and it is a polymer of nucleotides. So say for example that the circles which are shown are the nucleotides, right, and the lines which are drawn are the bonds which are connecting the nucleotides. So how will you define DNA? DNA is a polymer. It is a polymer of the monomeric unit nucleotides. Okay. So this single bead-like structure which we have drawn is a single nucleotide, and this line which is joining is a bond which is called as phosphodiester linkage. So now we come to know that DNA is a polymer which is made up of nucleotides, innumerable nucleotides which are linked by phosphodiester linkages. Okay, now we come to the circle which we have drawn here for to represent nucleotide. Its actual structure is shown in this figure. Okay, so a nucleotide is basically made up of three components. Let us one by one classify the parts of a nucleotide. This is the phosphoric acid which is present in a nucleotide, right? Then the second part is the sugar, this part which we are highlighting in green. It is the sugar, and the nitrogenous base is the part which we are highlighting in purple. Okay, so these are the three parts of a nucleotide, and let us first name them. The nucleotide is basically made up of a nucleoside and phosphoric acid. So the three parts are again clubbed in the form that two combine to form a nucleoside. So a nucleoside is a sugar. Okay, this is the first part, and the second part is the nitrogenous base. So 1 plus 2, that is the sugar and the nitrogenous base, they form the nucleoside, right? This reaction. And when a nucleoside is joined with the third component, that is the phosphoric acid, then it is called as a nucleotide. peptide. So I think it's clear that the single monomers which are shown in the polymeric representation over here, so this is a structure having all the three components, out of which you have to remember that the skeleton or the backbone of the DNA is made up of sugar and phosphate. These two molecules are involved in the backbone. The nitrogenous base is projected inward so that it can bond with the second strand of the DNA. As we already know that DNA is double strand. Now we come to one component in detail. The first component is the sugar of the nucleotide, right? So the sugar which is present in the nucleotide is a pentose sugar. Okay, and this pentose sugar has 5 carbons, and in numbering, see here, 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. So it is numbered from your right towards your left, and this is the second carbon, the third carbon, the fourth carbon, and the fifth carbon. Okay, this is important to understand because in DNA we will come to a slide where we give a name to the ends, the free ends of the DNA. So if the fifth carbon, let me highlight over here, the fifth carbon, if it is this one, if it is free, it is not bonded, then this will be called as a 5 dash end, or it is called as a 5 prime end if it is free. Okay, and if the third carbon, let me highlight this also, if the third carbon over here, this part, this OH is free, it is not engaged in bond formation or linkage with any other molecule, then we say that the C3 carbon, so this is called as the 3 prime or the 3 dash end. This we will require for anti-parallel nature. So you just remember for the time being that the sugar has two carbons which are very reactive. One is the fifth carbon and the second is the third carbon. Okay. Now we come to the second component. The second component is the phosphoric acid, and this phosphoric acid is in form of phosphate, that is iron, which is linked to the ribose sugar at the fifth carbon. Okay, so here two carbons are important. This is the fifth carbon and this is the third one, right? So this fifth carbon here, it is linked to the phosphoric acid. So this actually forms the backbone, the ribose sugar and the this one phosphoric acid together, they form the backbone or the skeleton over which the structure of the DNA relies. Now we come to the third component, the nitrogenous bases. The nitrogenous bases are, these are the nitrogenous bases. The nitrogenous bases are of two types, okay? The purines and the pyrimidines. The purines are double ringed compounds. That means their structure has two rings and they have either adenine or guanine. And pyrimidines are having three types, that is cytosine, thymine, and one is not given over here because it is not present in DNA. It is called as uracil, which is present in RNA, right? Now we come to the pairing. It is a rule which is always followed in DNA, that is a purine, let us write Pu for purine, will always pair with Py, that is pyrimidine. It will never happen that a purine pairs with purine or a pyrimidine pairs with pyrimidine, right? So the pairing of purine and pyrimidine is always fixed, right? The purine, if it is adenine, then it will be linked to thymine with a double bond, and if it is cytosine, then it will be linked with guanine with a triple bond, and these bonds are hydrogen bonds, right? So basically the nitrogenous bases, I'll just show you over here. What is the function of this base pairing? The nitrogenous bases, they are complementarily present in both the strands. Let this be our DNA. Okay. This is the helical nature, and so the bases, they project, and in this way the bonds link and stabilize both the strands. Okay, you are getting it. So the function of nitrogenous base is to form a hydrogen bond with a complementary base pair which is present on the opposite strand. Say for example, here if it is adenine, then here the first base will be thymine. Okay. If here it is guanine, then the second base will be cytosine. So always this complementarity is maintained. So the types of nitrogenous bases are adenine, guanine, cytosine, uracil, and thymine. As we already know that these are double-ringed purines and these are single-ringed pyrimidines, where uracil is present only in RNA and it is not found in the DNA. Now we come to the linkage. Now you just concentrate on this figure here. See, this is the sugar which we were talking about. This is the third 3' carbon which will be reactive, and this is the fifth carbon which is reactive. So you're finding that in this strand, this fifth carbon is free. What is the meaning of free? That it has a free OH group. It is not linked to anything else. Okay, so this end is free. So if the end has the fifth carbon free, then this end will be called as the 5' end or 5' end. Okay, and now we will just concentrate on this. See here, this is the nitrogenous base, this is the sugar, and this is the phosphoric acid. So the fifth carbon is linked to the third carbon of the previous nucleotide with the help of the phosphate. So there is a bridge, and this bridge acts as a link between the first nucleotide and the second nucleotide, and this linkage is called as phosphodiester linkage. So these were the basic skeletal structures which were very important from the point of view of structure of DNA. Now we come to the basic points which were very important in Watson and Crick model, and that those points were, the first nature which they explained was that it is a double-stranded what to say molecule, and it had a, see, this was, they proposed that it was a double helix structure like this. So these were the two strands, and they were parallel to each other. Okay, so the second point was that they were parallel, but then the direction was opposite. Like if this was the 5' end for this strand, then here it was the 3' end, and the second strand has, this as 3' and this as 5' end, right? So this nature of opposite direction, that if this strand goes in this way, the second strand has an orientation just opposite to it. So this is called as, so this strand is 5' to 3', then this strand will be in this direction. This nature is called as anti-parallel nature. Apart from this, one more important characteristic was explained, that there is a major groove and a minor groove in a DNA at one point, right? So at a single point there will be always one major and one minor grooves, and on one side there will always be, see over here, this is a minor groove, then there is a major groove, then again there is a minor groove, again there is a major groove. So alternatively on one side there is one major and one minor groove present, and the length of one turn, it always involved, say for example this is a single turn from here to here, this involved the distance of 34 Armstrong, and it had 10 nitrogenous base pairs. Okay, so how many base pairs were present here? 10 base pairs were present, and these base pairs which were present occupied therefore 3.4 Armstrong of space, and the diameter of the DNA molecule according to this model was 20 Armstrong wide. Right. So these were a few statistics which were proposed by Watson and Crick model. And this is a three-dimensional structure where you are seeing the skeleton. This is the skeleton you are seeing here. This is made up of sugar and phosphates. And the projected ones which are showing linkages in between the two strands, holding the two strands in helical orientation, are the nitrogenous bases which are bonded by hydrogen bonds. So with this we complete the structure of DNA, and I hope this module has helped you to understand the structure of DNA. Thank you.